Hello, good morning. Happy Friday the 2nd of October 2020. Here is a lesson about split brain research. As a disclaimer, I've tried to source out all the appropriate copyright issues on this. If anybody's spotted anything that may need to be removed for copyright reasons, then please let me know and I will edit appropriately. Okay, so something to kind of get you up and running with split brain research. Previously, you should have looked at the different areas of the brain. The motor area, Broca's area, the somatosensory area, the auditory area, Wernicke's area, and the visual area. What I'd like you to do for the first part is to note these down and match them to the appropriate definition or function. As an extension, you could consider why the areas have been colour-coded in that way. So please pause the video and then when you are ready, move on to that next slide for your answers. Okay, so going through your answers, the motor area is responsible for regulating movement. Broca's area is responsible for speech production and is in the left hemisphere, and what I will refer to as the study of tan by Broca, somebody who had a lesion on Broca's area and could only use the word tan. The somatosensory area sits behind the motor area and that is responsible for sensory information such as touch. The auditory area is an analysis of speech-based information. Wernicke's area is in the left hemisphere and that is chiefly for language comprehension. And the visual area is in the back of the brain in the occipital lobe, and it receives and processes visual information. Something that I'd like to point out is the colour codes correspond with different lobes. So the motor area and Broca's area is in the frontal lobe. In terms of the somatosensory area, that is in the parietal lobe. The auditory and Wernicke's area is in the temporal lobe, and the visual area is the occipital lobe. In terms of this PowerPoint, by the end you should be able to apply knowledge to answer past paper questions on split brain research, and this is lesson 4 out of 12 in the sequence. You should also be able to identify what hemispheric lateralisation refers to, and that is towards the lower end of the grading system. You should be able to describe the findings of split brain research. And finally, towards the top end, you should be able to evaluate findings related to split brain investigations. So the first thing I'd like to talk about is the idea that the brain is split into two halves. And this is what hemispheric lateralisation is. It's the idea that the brain is two halves and have different functionality. Okay, now historically there's been lots of different research into different lobes of the brain, which is something that we'll look at. It has been shown that certain processes and certain behaviours are controlled by one hemisphere rather than the other. Okay, so the hemisphere is important when we have a look at certain processes. So what we're going to have a look at, we'll have a look at patients with split brains. Now you may be familiar with the video of Jodie as a child who had half her brain removed to prevent epileptic seizures. Okay, and what Jodie will have had, she'll have had a commissurotomy, which is where the corpus callosum has been removed. Okay, and the corpus callosum is the thing that brings the two hemispheres together and the connecting fibres as well. One of the most famous people who's done work on split brain research looking at hemispheric lateralisation and the mental processes is Roger Sperry, and in 1988 he was given the Nobel Prize for his work. Okay, so what Sperry did, most of his work was with Michael Gazzaniga, and Sperry devised a procedure where what was happening was he was projecting images specifically into the left visual field. Okay, so left part of the visual field. Now that left part of the visual field is processed by the right hemisphere. Okay, the reverse is true. Okay, so what happens is the information comes into the left visual field, and the part of the brain that's chiefly responsible for that is the right hemisphere. Now, what he did was he wanted to have a look at 11 patients that had had their corpus callosum removed. All these patients had previously had epilepsy, and what he wanted to do was compare that with a control group. And what he did was he compared it with a control group who had a corpus callosum. Now, in 1967, Sperry and Gazzaniga performed research on split brain patients. Now, what he did was, what the pair of them did is they set up a system to, again, transmit information into the left visual field, which is processed by the right hand side of the brain, the right hemisphere. So it was really important he had a carefully crafted experiment, okay, and it's make sure that it was the methodological processes were valid. So have a look at the diagram on the screen, pause it if you feel like you need to pause it. So clarification there that split brain patients have undergone surgery to cut out the corpus callosum, and previously they were having epileptic fits and seizures. So if there is input into the left field, that is processed by the right hemisphere, and vice versa. So if there's input from the right field of vision, that is processed by the left hemisphere. And what happens, and I'm in the central image now, is a word was flashed very briefly to the right field of view, okay, and the patient was then asked what they saw, okay. Now, because the left hemisphere is dominant, then the patient's answer matches the word, okay. So the fact is, in terms of only going into the right field of view, if the left hemisphere was present, then they'd be able to match the word. Now, if the same word was flashed into left field of view and that patient was wearing an eye patch or a blindfold, then the right hemisphere would not be able to share information with the left. Therefore, the patient would be unable to say the word. However, the mental processing for drawing it and processing is still available, so therefore they would be able to draw it, which makes us conclude that the verbal side is on the opposite side of the brain, but a side for mental processing must be on the correct side of the brain and able for them to draw it. So they wouldn't be able to say it, but they would be able to draw it. So in terms of the procedure, there was a stimulus, and the stimulus in this circumstance was a word, and the word was face. And that was being displayed to the right visual field, and that's processed by the left side of the brain. Alternatively, you could do it to the left visual field, and that would be processed by the right side of the brain. So after the stimuli had been presented, the participants were asked to state what the stimuli was and what had been presented to them. Participants would have one eye blindfolded to prevent them moving their heads, and the stimuli entering both visual fields, okay, therefore being sent to both hemispheres. So it's controlled by making sure they only went into one visual field. The other thing is, the stimuli were presented for 0.1 seconds, so 1/10 of a second. This was so that the patient or the participants rather would not be able to move their head and try to anticipate what was going on. So, two key things that there are to, when you're trying to get to grips with this study, things that you need to understand. The fact is that the right side of the brain deals with information from the left side of the body and vice versa. The other key thing to understand is the left side of the brain is responsible for the understanding and production of language, which refers to Broca and Wernicke's area. Okay, if you've got access to the PowerPoint, because I've sent it to you, then please press the video to watch Sperry and Gazzaniga's study. If you don't have access to the PowerPoint, please look at Sperry and Gazzaniga's studies on YouTube. Right, okay, so in an exam situation, you may be asked to explain what the findings show. So, what I'm going to do on this slide is I'm going to read to you what it says in the findings box, and on the next slide would be an explanation. Please pause the slide and attempt to do this yourself beforehand. So, in terms of the description of what the participants saw, if a patient was presented, if a picture of the object was presented to the right visual field, the patients could describe what they had seen. However, if it was presented to the left visual field, they could not. Okay, and then pause the video and explain what these findings show. Secondly, recognition by touch. When presented with an object in their left visual field, although they could not verbalise what the object was, they could find a matching object from a grab bag with their left hands. Okay, and again, please pause the video and do an answer for that. There is a typing error on this. Okay, please accept my apologies. For composite words, if two words were presented to people at the same time, one to each visual field, for example, key to the left and ring to the right, the patient would write key, however, their left hand would say ring. Okay, so they would say ring rather than key. Okay, so that's the typo. I have corrected it on this slide. So in terms of your explanations, language is processed in the left hemisphere. The patient can't describe objects in the left visual field because it is processed in the right hemisphere. And because that participant has no corpus callosum, hemispheres can't communicate. In terms of recognition by touch, the patient would not be able to verbally identify, and again, this is due to a lack of corpus callosum. However, they would be able to understand what the object was using the right hemisphere, and therefore they'd be able to understand and select the correct object. Finally, the reason why they would say ring, the reason why is key went into the right side of the brain, and this controls the left side of the body, and it can be written with the left hand. Ring went to the left side of the brain, and that controls speech, so they would be able to say that word. Another thing to consider is in terms of matching faces, and we'll have a look at the Mike and Me study by Turk et al. When asked to match a series of faces, the face seen in the left visual field was selected. So here you've got a matching face image which is made of two different people. So when composite of two halves of a face was split between the left and right visual field, the half seen in the right visual field dominated in terms of verbal description. However, that seen in the left visual field was better at finding the matching face. So one side's better in description and one side is better for matching up. So the left hemisphere dominates in terms of verbal description, the right hemisphere dominates in terms of selecting a matching picture. Okay, so in terms of the three evaluation points here. Number one, explain why it's difficult to make generalisations. Something to point out is it's an unusual sample of people here. What you've got is a selection of people who have previously had epileptic seizures. They've had their corpus callosum severed, and only 11 people took part in the original procedure. Okay, so in terms of making generalisations to the whole population, okay, this is a small sample and it's an unusual sample of people. The other thing is, Sperry's control group of 11 people with no history of epilepsy, this is inappropriate because it's the epilepsy that may have caused a neurological change and not the severed corpus callosum. Finally, brain plasticity may be an argument against Sperry's findings because functional recovery may occur. So it's the idea that the neurological plasticity may change what happens with the participants. So for example, with Phineas Gage being able to drive coaches around Chile, eventually he will have undergone some plasticity following his incident with a railroad spike. So, in terms of the methodological issues, what I'd like to do now is pause the video and explain why each of the following are problems for split brain research. So your problems are that some participants had received drug treatments since their corpus callosum had been severed, the disconnection between hemispheres was greater in some participants than others, and the comparison group had comprised of people who had not had epileptic fits. Please pause the video and answer these questions. Okay, so some participants had received drug treatments that could have had an impact on their brain function. The other thing is it could have also affected their verbal and visual abilities, and this means that behaviour in research may not only have been influenced by split brain. The fact that disconnection between hemispheres was greater in some participants than others means that there may have been some communication between the hemispheres. However, for some people there may have been no communication. Therefore, some patients may have been more split brain than others because not all of the corpus callosum was removed. And finally, the comparison group that had comprised of people who had not had epileptic fits. The comparison group differed in two ways. Not only were the control group not split brain patients, they also had no history of epilepsy. Therefore, epilepsy is a confounding variable. Epilepsy may have been another factor that influenced the responses of the split brain patients, and this will have not influenced the control group. In terms of the Mike and Me study by Turk et al., to participant JW, what was presented was a mixture between the researcher's face, Michael Gazzaniga, who is named as MG in this, and a mixture of their own face as JW, going from 90% of JW to 10% of JW, and a mixture being in the centre. and split brain experiments were performed to see which JW would recognise. Okay, what I would like you to do is consider the ethics of the study. What are 5 ethical issues, or 5 ethical issues in psychological studies in general? Explain the main ethical issues that was being upheld in this research by Turk et al. And then explain how Turk could have dealt with one more ethical issue in this research. Please pause the slide and attempt that question for me, please. Okay. It should say 3 ethical issues. Please accept my apologies. There may be 5. Well done if you've spotted 5. Okay. Confidentiality is 1. Protection from harm is another. And the participant's right to withdraw is another. Okay. So explaining the main ethical issues. Confidentiality is important. So here the participant has been referred to as JW rather than name. Protection from harm needs to be considered with this. His image, JW's image, is available. Okay, could this lead to the participant's harm later on? And also the right to withdraw. Has participant JW been informed that he can withdraw at any time? So one of the ethical issues, referred to as JW, needs to remind participant before and after they can withdraw for a 2-week window after, and it needs to ensure that the participant does not come to any more harm in the experiment than they would in real life. Okay, if we're evaluating Sperry and Gazzaniga, which was the original, it's too simplistic. So the researchers oversimplified the roles of the brain, suggesting that different hemispheres have functions. So what this means is the concept of hemispheric lateralisation may be too basic an explanation for brain function. So it suggests that the left hemisphere is responsible for language. However, we know that language may involve planning, responding to visual input and responding to emotions. So it could be argued that language is too complex to be completely explained by the activity only in the left hemisphere. In terms of methodological strengths, a good thing about this research is it's been really controlled and it's a good example of standardisation across the 11 participants. See if you can explain this point more by pausing the video before you have a look at the explanation points. With regards to the explanation points, for example, they were able to control the visual field that the participants saw the image or word in, therefore it was use of a fixation point. So this meant that they can guarantee via the use of a blindfold that they were able to make sure they only went into the left visual field, for example. Also, images were only presented for 1/10 of a second. This meant that Sperry and Gazzaniga were able to accurately test the impact of having a split brain, and it made the research a valid test. They were not able to move their field of vision in time to incorporate the stimulus in any other way. One of the notable split brain people, however, is Kim Peek. Kim Peek is who the 1988 film Rain Man is based on. Okay, please, if you don't have access to my PowerPoints, please research Kim Peek using YouTube. Kim Peek has bilateral hemispheres of the brain. He has no corpus callosum and he was born with severe brain damage. What happened with Kim Peek is that essentially his 2 brains operate independently, or 2 hemispheres of the brain operate independently. So for example, this means that Kim Peek has managed to read 12,000 books very quickly, and he can scan a page in a matter of seconds by running both of his eyes down the page. Okay, this is an example of neural plasticity and how he is developed differently. He has been suggested that he is a savant. Originally, he was diagnosed with autism until the discovery of the corpus callosum absence was found. Please watch a video about Kim Peek. Okay, here's another image. So in terms of all split brain patients that were studied in this study, all the patients were right-handed. Okay, and when presented to the different visual fields, you can see the difference in drawing, okay, which suggests that the different hemispheres of the brain are responsible for interpretation of information and drawing. Finally, what I'd like you to do, again, if you don't have access to the paper, please pause the video and now complete 6 marks of split brain exam questions. Question 1, split brain patients show unusual behaviour when tested in experiments. Briefly explain how unusual behaviour in split brain patients could be tested in experiment, 2 marks. And number 2, briefly evaluate research using split brain patients to investigate hemispheric lateralisation of function. So that's an evaluation question, 4 marks. Please pause the video and attempt before going through the answers. Okay, so for the first one, what you need to do for 2 marks is a clear, brief explanation of an experimental procedure. Okay, so suggestions. Okay, split visual fields. Okay, or dichotic listening, which is referring to audio experiments, which is an alternative. A plausible stimulus, such as visual, faces, words, an auditory example, digits or music, and a plausible task for the patient, such as verbal, visual spatial response, drawing, matching, etc. That's question number 1. Question number 2. For you to be in the 3 to 4 marks level 2 bracket, your evaluation needs to be relevant and well explained and focus on the usefulness of split brain research for the study of hemispheric lateralisation. For level 1, evaluation needs to be relevant, although there's a limited explanation and the specialist terminology is not always used appropriately. So your key points for this is the disconnection between the hemispheres was greater in some patients than others. Some patients had experienced drug therapy for much longer than others. The comparison groups were not considered to be valid as they were often people with no history of epileptic seizures. The data were artificially produced, as in real life a severed corpus callosum could be compensated by the unrestricted use of 2 eyes. Okay, therefore a split brain patient is not walking around with an eye patch, so it's an artificial situation. And the idea that research is related to small sample sizes. Thank you very much. Thanks for listening. If you have any questions, please feel free to get in touch. All the very best.